And then I'll talk a little bit about how you design a study that is good enough. But the truth is, you kind of need like a whole class to really know how to do the good enough study, but you don't need a whole class to understand why these simpler things are not enough and why you need to go further. And so I'm going to talk about that mostly. Okay, so I want to talk first about disease clusters. And so you might know a little bit about disease clusters already because I just talked a little bit about a little fake example one. A disease cluster is a group of similar disease cases that occur in a defined population, a geographic area or time. They're often investigated by state or local health departments. As an academic, I've never investigated one. Um, and there are some real problems with whether you can really associate any exposure, like figure out what caused a disease cluster. And some of those problems are how long people have lived or worked in the area, and whether they all have certain risk factors that cluster together, like people live and work, birds of a feather flock together. So people kind of live and work and they may share traits. And I'm going to talk a little bit about random distribution in space and time. And I'm talk a little bit about this Texas sharpshooter analogy to kind of get an instinct for why disease clusters but frankly, they're not usually solved. um, but they do take a lot of time and effort. Okay, so the example of a real life disease cluster that I'm going to talk about is here in California. Does anyone know California or Marin County at all? No. Okay. So Marin County is the county just north of San Francisco. And so it's across the Golden Gate Bridge and it's just north of San Francisco. And it is a very wealthy, very white county, very rich. It's lovely. It's beautiful out there. It's gorgeous. And it's sort of a working suburb of San Francisco. People do commute to San Francisco. But it's like the doctors and lawyers commute to San Francisco from Marin County. So it's a very wealthy county. Um, and a very desirable location. Marin County, about a while ago now, maybe about 20 years ago, had a spike in their breast cancer rates, and it made the news. And some health agencies stepped in. So San Rafael is one of the big town in Marin County. And this is, their paper. Their four national health agencies will try to answer what's mystified Marin County residents for years. Why does this community have what appears to be the highest rates of breast cancer in the nation? And this continued to make other news. Marin has the highest breast cancer rate, 20% rise in 1 year is a mystery. Okay, well, that's scary. 20% rise in breast cancer. This has even made the international news. This is the British newspaper, California's breast cancer plight. And you can see this like trend line. Marin is going to go like way off the charts. Okay. There was a big uproar about the breast cancer um, rates in Marin County. And a lot of attention paid, a lot of work went into it, and they use cancer registries. They really tried to figure out what was going on. But sort of the first question of what was going on is there really even an increase, right? So that first question is, do we think, like in the Peach State Community Center, like, is it just kind of, is it actually more than the background rate? I mean, that's really the question, right? Like, is there really a rise? Is all of California seeing a rise? Like, first, you kind of want to get a lay of the land. And so this chart was put together by folks who studied breast cancer in California. And what this chart says, so this is now age-adjusted rate, so we don't have to worry about if the women are older or younger in Marin County. This is over a pretty long stretch of time, 1988 to 2013. Marin County is this up and down blue line. The Greater Bay Area, so there's like five or eight or something counties that all touch the bay. That Marin County is one of them. San Francisco County, where I live, Alameda County. There's a bunch of counties, and so all of these counties are here in red, and then all of California, but not Marin, is this thin black line here. So looking at this, what do people notice?
Compared to the others, like California Greater Bay Marin County is like really turbulent.
Marin County is really what?
Like turbulent. it's not stable like the others, but not might be because we're looking at like a smaller.
Right. That's exactly right. It bonks up and down. It's going all over the place. And you're right. It probably is because it's smaller. So has anyone taken a statistics class or a data science class? So Jen, have you? Yeah, you have. So that's why they notice the small sample size. And so when you have not very many people, you can get these like weird things can be high, things can be low, things can be high, things can be low. And it's just because it's like unstable, basically. Like the denominator is sort of unstable. But if you look sort of over time, then it kind of seems like, well, maybe the middle line is sort of the Greater Bay Area or California, right? Like it's bonking up, but it's also bonking down and then up and then down, right? So it's sort of going all over the place, but on average, maybe it's kind of okay. Maybe it was a little higher here and maybe it's a little lower there, but if you really looked over a really long period of time, that might also even out. And so this is sort of the first trouble with figuring out clusters is that the clusters usually occur, I mean, they do occur in a small geographic location, a county, a town, a workplace, something, and you're very unstable. It's very unstable. It's very hard to figure out what's going on. And is there, if it's difference, is it just like bad luck or is it a real difference due to something? And so that's one thing. But what people notice is that, and then the California and the Greater Bay Area to me look pretty similar, frankly, right? What happened here, and when people got super alarmed in Marin County was right there. This was the year, right? Raised the red flags, called in all of the health agencies. And frankly, it was a pretty bad year. I'm going to give them that, right? Like it had climbed a lot and it's not clear where it's going. But at that moment, it turned out it was not going to stay that high, right? But you can't, it's hard to see the future. So you don't really know. So when you take a look at these really rising incidence rates, it's a little unclear. Like, is this just like a weird spike that's going to go away and doesn't really mean anything? Like, it's not really that there was a ton more breast cancer this year than last year. Maybe a lot more people went to the doctor and got screened. Maybe they opened a new clinic and or something, right? Like it's you kind of want to see where it's going to go over a period of time. On the other hand, people are getting diagnosed with breast cancer, and breast cancer is like scary. And so no one wants to be told like, let's wait 5 years and see what happens. So this tendency, but what people don't do that in clusters, right? They see a rise, they notice there's a cluster. Right now, there's 10 new cases in my community. Right now, there's 5 new cases in my workplace. And this tendency to focus on that rise is what's been called the Texas sharpshooter problem. And the Texas sharpshooter is this sort of stories like I mean apparently there's a legendary Texan and he would fire his rifle randomly into a side of a barn and then after he fired his rifle he would walk up to the barn and he would draw his target and then he would invite all his friends over and say, look, I made the center of the target each time, right? It's sort of this post hoc drawing of the target around what happens to be there. And that's sort of the analogy people use for clusters a lot. Like we're not really looking in every community that has high radon to see what's going on. We're just looking at this one community that has a spike and we're making a big deal out of it. But that's not really looking at the whole picture. And so it's that's why almost every single cluster that's investigated, which takes a lot of time and a lot of effort, turns out to kind of there's no there. But a lot of time and effort does go into it. The other things to consider are for something like cancer. I've said this a couple of times, but people's whatever exposure causes breast cancer happened 10 or 15 years ago. So if you're thinking about a bunch of women in Marin County who are getting breast cancer, well, where were they living 10 or 15 years ago? Have they always lived in Marin? Are they longtime Marin residents? Did they recently move there? They recently moved there. There's nothing about Marin that caused their jobs. And then you want to think about I know y'all don't know Marin, but you might know other wealthy white areas. And does anyone know any other risk factors for breast cancer off the top of their head? You're not a bunch of breast cancer epidemiologists. It's fine. So, Daisy, do you know?
Yeah, night shift that we talk
Oh
about.
yeah, working night shift. That's been associated with breast cancer. So that could be possible if we had a bunch of night shift nurses living in Marin, we might want to worry about that. But a non-occupational risk factor.
Oh, if you don't have any kids, that it
Yeah.
increases more. Yep.
Right. So having kids is protective, having kids younger is protective, and breastfeeding is protective. So having kids later or not having kids or not breastfeeding all increase risk of breast cancer. And so when you're looking at wealthy communities in general, but especially wealthy white communities, these are mostly a larger percentage of these women have delayed having children. So they're having kids later, which I mean, I did too. So I'm not saying this as like a source of judgment, right? But compared to the rest of the Bay Area, you might have a higher portion of very well-educated women who waited until after law school to have their kids, and that just puts them in a bracket where they're maybe more likely to get breast cancer. Um, there were some very unhinged theories that went around at the time that were particularly hard to stomach from my perspective. So the other thing about Marin County is that it's north of San Francisco and it's west, right across the bay from Richmond, which is a very poor city, non-white city, and has an oil refinery there. But the weather pattern in the Bay Area is that the wind comes from the ocean, comes over Marin County and comes into Richmond. I mean, because the wind comes from the ocean. I mean, that's just where the wind comes from. But they all like had this, there was a group of people that had this theory that somehow the oil refinery pollution was going backwards against the wind to get the women in Marin County, but somehow not the women in Richmond. The whole thing was a little bit bananas. But it made a lot of press that like this refinery was somehow targeting them. Because so at the end of the day, it turned out, I had the slide. So the detective sharpshooter is a tendency to assign unwarranted significance to random data by viewing it post hoc, so like after the fact, in an unduly narrow context. This is human nature. Like, I know I'm not mad at them. So it turned out, a few years later that they realized there was no breast cancer rate in Marin. We say the culprit's not environmental, but the residents don't agree. So Marin didn't want to take notes. Now Marin is no longer a hot spot for breast cancer, in 2015. And now there's a new question. Marin's breast cancer rates have plummeted. Why? Okay, so you start like they're going to like spike sort of randomly. They're also going to come down randomly if the average is sort of there. And so the story sort of fizzled out, as often happens with clusters. But I would be wrong to say there's never been a useful cluster. There've actually been some extremely clusters that have provided a ton of information. And so two very famous examples are in the late 50s and early 60s, there was a medicine that they were given pregnant people that would help them sleep that unfortunately turned out to be a teratogen, which means it's a chemical that harms the fetus when it's in utero. And so a whole bunch of these babies were born with these shortened limbs, and people were like, stop, what's happening? And they were able to sort of go back and look in the medical records of the mothers of these babies and realize that they had all been prescribed this sleeping medication. And so that's a cluster, but not a red herring. In addition, our first understanding of what HIV/AIDS was found in a cluster of pneumonia that they couldn't treat. That these previously healthy gay men got pneumonia and they just were dying from pneumonia. Like they weren't responding to any of the treatment for pneumonia. And it was from these 5 cases that we first realized what HIV/AIDS was. So it's not every cluster is silly, but most clusters are really are not very useful. Okay, the next study design I want to talk about that's very common is our ecologic studies. So ecologic studies are a kind of study that correlates group measures of disease with group measures of exposure. And this is super common. It's pretty affordable. You can often do it without even getting access to personal level data. Like, I mean, you could download some data from the internet and do one of these studies if you knew how. And so they're very accessible, they're very cheap, they're very common. And they're somewhat helpful, but they're not perfect. So some examples that I thought of, but there's a million examples. You could look at average SNAP is food like food stamps, like I don't know what they like a government assisted food like credits in stores. So poorer folks will get government assisted food, and if in the areas where they have the SNAP system, are there lower birth rates? So does giving people extra money to buy food help with low birth rates? Does average vaccination uptake reduce COVID death rates? So in a county, if more people are vaccinated, do we see less COVID? These are reasonable questions and easy to do. We have average air pollution in California counties and asthma rates in these counties. Again, lots of studies just like this. And so I sort of want to unpack, although it seems totally logical, like why wouldn't this work, especially if it's cheap and easy? I want to talk about a very sort of famous example that looks at exactly where the problem can occur in these studies. And so this example is not really environmental epi. Has anyone taken a sociology class and know this example? No. Okay. So Emil Durkheim was a sociologist a long time ago in 1897. And he was interested in looking at the relationship between religion and suicide rates. So he had some theories about Catholicism versus Protestant, which were the two predominant religions in the area he was from in this Parisian provinces, which is like where Germany is now. And he thought there was something about the social structure of Catholicism versus Protestantism that was more alienating or more community-based and would really have a role in suicide. And so he did a study and he looked at suicide rates in a bunch of these provinces, which are like counties or zip codes or something like that. And he saw that the higher the more people in the county that were Protestant, so this would be like 20% of the people in the county are Protestant, 40% of the people are Protestant, 60% are Protestant, 80% are Protestant in the county. The more of the people in the county that are Protestant, the higher the suicide rate. So what would you conclude from this?
Like the more religious you are, the higher probability.
Yeah, or specifically the more Protestant you are.
Yeah.
Right? And so you might think like, oh my gosh, we better get our act into gear and go into some of these Protestant churches and like help people, right? Like we need to do some anti-suicide education in Protestant churches. That's what I would want to do. If I wanted to do something about it, that's what I would think. But, and that is actually what he concluded, right? That Protestants are more likely to die from suicide. But let's think about this for a second. Do we have any information on individual people in this study? Like he had percent Protestant in the county and suicide rate in the county. So there's actually a major leap that he made and that I sort of encouraged you to make. So I'm kind of like dug a hole for you and then like pushed you in it. So don't be upset about that, sorry. Which is that we're assuming that these suicides are happening among the people who are Protestant, but we don't know that. What we know is that the counties with more Protestants have more suicides. And those are not necessarily the same thing. They sound like they're the same thing. But the fact that they're not the same thing is exactly what we call the ecologic fallacy, the reason why these studies sort of fall apart. And so here's what really happened. Here's what really, someone dug deeper and looked more at the data. So none of these regions was only one religion. People were either Protestant or they were Catholic. I think there were only two religions. I don't really know. That's the story as I understand it. So you could ask the question, was it really the Protestants who were dying? And it turns out it actually wasn't the Protestants who were dying. It was the Catholics in the heavily Protestant areas who were dying from suicide. That's totally different, right? That's a very different story about who's dying. And it gets into, minoritization and alienation And yeah, things that sort of make a little bit more sense in our understanding of mental health and suicide, and what it means to be a minority group. So in these heavily Protestant areas, more people are dying, but it wasn't the Protestants. It was the Catholics in these heavily Protestant areas who were dying. And that leads to a very different conclusion. Now, suddenly, I might go to the Catholic churches in the heavily Protestant areas and figure out, like, how do we help people, right? Or go to the Protestant churches and be like, yo, stop being all mean to the Catholics and include them in your parties, right? Like, that's a very different intervention because it has a very different meaning. And so when you look at something like this, you know, the conclusion is now that the non-Protestants in heavily Protestant areas are more likely to die from suicide. So when we think about these studies that have group level exposures, like percent Protestant, percent air pollution, percent SNAP use, and group level trends, percent asthma rate, you know, asthma rates or preterm rates or something like that, you have to really stop and think, like, okay, this is probably true. The higher the exposure is, the more outcome there is, but it does not tell me that the exposure caused the outcome, right? It does not tell me that Protestant caused suicide. I want it to tell me that. That would be very convenient, but it does not tell me that. And so, assuming that these group level, anyway, so that's a problem. So when you see these ecologic studies, which are very common, you know, stop and think, what is it really showing? And is there a leap that our brains are making that that's unwarranted? Okay, so now I'm going to spend my last half hour talking about the right way to do things, which is cohort, cross-sectional, case control. I'm not really going to talk about hybrid designs. I'm not really going to talk about experimental trials. Okay, so cohort studies define a group of people. It could be lots of people. It could be not very many people. That doesn't really matter. And they follow them through time to observe disease occurrence in relation to whatever their exposure was. And so here is a little schematic of what we call a prospective cohort. And the difference between a prospective cohort and a retrospective cohort is just about time. It's not really about the design of the study. So in a prospective cohort, an investigator defines a population of people. You know, I can say, like, you know, a bunch of people who were hired in a factory. And then I figure out who maybe I'm studying for cancer. So first I have to make sure that anyone who already has cancer is not, I'm not going to follow them to see if they get cancer because they already had cancer. They're not at risk to get cancer again. And then I figure out who's exposed to some chemical and who's not exposed to some chemical. And then I wait 40 years and I find out who gets sick and who doesn't get sick from this chemical. So this is a disease and not disease, not disease and disease. That's what the D's and the N's mean. And so that's a prospective cohort. So you first identify a cohort, you follow them into the future, you collect data on both exposure, and then you wait, and you collect data on disease outcomes. And it's really the gold standard of observational epidemiology. This is like what we think is the best way to do it, because you can get very high quality data. But I was going to ask you what the problems with it are, but look, it says here, right here on my slide, they're very expensive. So if you want to study a rare disease like lung cancer or Parkinson's or something like that, you need to have thousands and thousands of people in your cohort. And then you need to ask them all questions and follow them and track where they live. And oh, did you put something in there about Emile Durkheim? That's funny. Yeah, he was a great sociologist. He studied religion a lot, so I only sort of picked and choose the pieces of his the point that I wanted to make about him. Okay, so and they can take forever. If I want to know if some exposure causes cancer, I'm going to see who's exposed, and then I'm going to wait like 40 years. I'm retired. My postdoc is retired. It's going to be a really long time until we know whether this chemical causes cancer. I don't want to wait that long. I want to know if the chemical causes cancer because I want to do something about it now. I don't want to be like in 50 years, like, oh, geez, we should have done something about that a long time ago because now we have like all these more people who are going to get cancer. Okay, it's a little bit of a problem. And that's a little bit why we don't do more prospective cohorts. They're not good for rare outcomes. But we do have some really famous ones. Now I want to talk briefly. Has anyone heard of the Nurses' Health Study? This is a completely brilliant cohort, and we have learned so much from this cohort. And so this is out of Harvard University, and it started in the 70s. And they enrolled, and they now have 3 waves. It's pretty complicated. But the first wave, they enrolled almost 250 nurses who were 30 to 55. They were married, and then they were all over the US, but in the 11 most populous states. So Massachusetts, Texas, California, I don't know, wherever, New York. And the original research question was they wanted to know about hormone use and diet and smoking and birth control pills and menopause. And actually almost a lot of what we do know about hormones in women and diet and smoking does come from these nurses. They collected biologic specimens. They did a bunch of subsamples in them, and they recruited nurses on purpose because they thought that nurses would be much more likely to accurately report their own health symptoms. And they could also do things like draw their own blood and freeze their own urine samples and mail them in, like really cool things that made the study possible. And they also thought they would be pretty similar to each other. Like there wouldn't be that many differences in SES, and they're all nurses. And so there are some tricky bits in there, but and they've been following them now for years and years and years. And just we know so much information from this cohort. It really is a great cohort. A retrospective cohort so prospective means going forward in time. That's what that means. Retrospective means going backwards in time. And so in a retrospective cohort, if you don't want to wait 50 years to find out who gets cancer, you can do a retrospective cohort, which is that you still have the same thing. You start with the population, you find out who doesn't have disease, you collect who's exposed and who's unexposed, and then you look to see who's diseased or not diseased. Except instead of the investigator starting here in 1976, the investigator can actually start like today. Like I can do this project now. I can say, okay, I've collected some data on everybody who's ever worked in this factory. And now I'm going to pretend like I'm back in 1976, and I'm going to look at the records, and I'm going to see who didn't already have cancer in 1976. And then I'm going to see what were they exposed to from 1976 until 2000. And then I'm going to follow them through time till 2025 and find out who has cancer, right? So you sort of construct the whole thing, but you do it retrospectively. And in fact, most cohorts are a little bit of a hybrid of this, where like I work on a cohort now where they started a retrospective cohort in the 80s. A group of people were hired at General Motors making cars in 1940. So in 1980, this was a retrospective cohort going back to 1940. But then we've prospectively followed them from 1980 until now. We're still following them to find out whether those chemicals made them sick. So you can have the bit of the time can get sort of messed up. So this is a pretty great way to still have all of the benefits of a cohort study, but not have to wait 50 years to find the answer. But it does mean you need to have very good records, or people need to be, you need to have some way to determine what their exposures were. So these are actually terribly expensive and slow, but they are faster and cheaper than prospective cohorts. And you can study lots of different causes of death and disease at the same time. And these are the most common type of design in occupational epidemiology, because you can sort of clearly define an occupational group, a bunch of people who worked in a factory, a bunch of people who were hired into a textile workers, if you can follow them. It's a little harder to do for environmental epidemiology. It's harder to say like everybody who lived in the US in 2000. Okay, have fun finding all of them. And then they move and then they leave. And then, it's like way easier to be like, okay, records at a company, that's pretty straightforward. We have a lot of really incredible occupational cohorts, and actually almost not every, but almost every human carcinogen that we know of has first been identified in an occupational cohort. So diesel exhaust, benzene, silica exposure, asbestos, have first been identified in these occupational cohorts of workers. They're just very valuable, even if us non-workers are also exposed to them in other parts of our life. Like diesel exhaust was first really shown to be a lung carcinogen in a group of underground miners, people who were working, underground in a mine, and the machines that were digging doing the mining and digging the stuff off the wall were these diesel powered machines, as the diesel's getting trapped underground with these workers, all day. And so in that scenario, they were able to show that diesel was a lung carcinogen. But of course, I live in a city, there's diesel exhaust all around me, right? And I don't work in a mine. And so that is, it translates to non-working populations too. I want to talk a little bit about case control studies and give a little instinct for why they end up being really functional. So in a case control study, you don't have the whole cohort. You don't need all thousands, 100,000 nurses, for example. You can do it with fewer people. And so if you think about what a cohort study is, with my little white and black dots here, this is the whole population. This is the source population. The white and black just means if they're exposed and they're unexposed, and some of them become cases, but not very many of them. And so I mean, these are not very many dots, but the Nurses Health Study has 250,000 dots. The cost of calling them all up and following them is tremendous. What if we didn't need to do that? What if we just needed to have all the cases and a sample of the cohort? And it turns out that from a math perspective, you get all the cases and you get a random, this is not really a random sample, but whatever, a sample of the cohort. It turns out that you can find the same answers with these fewer people than with all of the people. You need all the cases, and then you can have just a subset of the cohort. And that can be great because maybe you only have enough money to interview a subset of the cohort, or you want to do like a blood test or something that's like totally cost prohibitive to do on 200,000 people, but you can figure out how to do it on 200 people. And so case control studies can be like a really valuable way to go. And this is called a nested case control study because the idea is like you had the cohort, but you pulled out just some of the controls for cost purposes to interview them. Most case control studies are not nested like this, and the degree to which they veer from this is the degree to which they're biased, which means they get the wrong answer. So in a case control study, you compare people with a specific disease, cases of whatever, to people without the disease, the controls, from the population that the cases arose from. And they're really good for rare diseases, and they're really common in environmental epidemiology because there's a lot of trying to figure out what people are exposed to, and you've got to talk to people and get that information. And so they're very common. But as it turns out, it's actually really hard to ask people to reconstruct their past exposures. And so if you think, that's one thing. So if you think about this, like if someone were to ask me, like I'm in their study and they're like, were you exposed to any pesticides as a kid? I don't know. Like I wasn't raised on a farm, so no. Except like we had slugs, actually, and ants, I guess. We had those ant traps or something. And then like my mom grew tomatoes in the backyard, and then once there were aphids everywhere, and I think she sprayed them with something. Okay, Sadie, thank you for answering that question. Which pesticide were you exposed to? Oh, I don't know, the one in the ant trap, and then the spray can, it was like green or something. Okay, how old were you? I don't know, like 2 or 10, right? This suddenly becomes like super terrible data, but you can't do a whole lot better than that, right? Like who really remembers all of these things? So it's really hard to reconstruct these past exposures for most people. Selecting the controls is hard unless you're really actually doing it nested in a cohort. It can go south really fast. And you get what's called, you can calculate the odds instead of the risks in a case control study. I'm going to skip that because I'm going to talk a little bit more about that later. So I said at first that these analytic studies all had some portion of time. And a prospective cohort study, you start with a group of people and you follow them into the future. A retrospective cohort study, you start with a group of people and you go back and calculate what their exposures were, then you follow them into the past, but you're kind of going backwards. In a case control study, you start with a group of people and you ask them questions about what they were exposed to. And in retrospective cohort studies, usually not asking anybody anything. It's still records-based, like job records or something like that. But in a case control study, you're asking about the past. Randomized control trials go forward. What I'm going to talk about next is a cross-sectional study, which really doesn't have any time. Everything happens at the same time. And so in a cross-sectional study, usually it's a survey of a group of people. We have NHANES, our what does NHANES stand for? National Health and Nutrition Exposure Survey. I don't know if I did that right. We have a lot of cross-sectional surveys, national cross-sectional surveys are very useful, but they basically, you get a survey and you're like, what'd you eat today? And what are you sick with? And do you have asthma? And do you have any pesticides in your house? And all these questions all right now. Okay. Which is great. I mean, people could do that. You get better quality information because I can look under my sink and tell you if there's any pesticides, or in the garage and tell you if there's any pesticides. I might remember what I ate today. I might not, but that's a different problem. And I probably know what I was diagnosed with, right, today. But the problem here is that, well, the good thing is that they're relatively quick, cheap, and easy to conduct. But all the data are collected at once. You can study multiple outcomes and exposures all at once. They're really good for descriptive analyses and really good for generating hypotheses, but the problem really is that we don't know whether the exposure came first or the outcome came first. And actually a lot of these surveys, they do also collect biologic samples. So like cheek samples or urine or something like that. And they can say like, oh, this person has really high amounts of aluminum in their blood, and they use deodorant that has aluminum in it. Therefore, we think the deodorant is causing these high amounts of aluminum in the blood. Okay, well, that's a reasonable hypothesis. Actually, maybe we should look into that, right? And this is a real example, but it turned out that actually once you started to look longitudinally, there was no there. That aluminum in the deodorant had nothing to do with aluminum in people's blood. But from this cross-sectional study, it's a little hard to tell, like, what's what. But it can generate generate good information, but it can't really tell you whether something caused something else. And in fact, sometimes with these biologic samples, and what really was happening with the aluminum thing is there are some people who didn't process the aluminum very well. And so tons of people have aluminum in their deodorant, but most people don't have high aluminum blood levels. But there are some people who have a metabolism problem, and so they're holding on to aluminum everywhere that they can get it, and that's causing a problem. But it's not quite the same problem as we think it is. It's like a different kind of problem. This type of study design is not great for rare diseases or diseases with short duration. I'm going to stop there. And you can get the wrong answer really easily. I have a little bit of time left, and I want to introduce a couple of things that you might see when you read research studies, and that's how we present our results. And so the most common way that we present our results is with this measure of relative risk. And all these words get super confusing, but that's just how we do it. So relative risk just means one risk relative to another risk. And almost always when people are talking about it, they're talking about a ratio. So one risk divided by another risk. So it's like a fraction. So we have risk ratio, one risk divided, risk in the exposed divided by risk in the unexposed, a rate ratio, the rate in the exposed divided by the rate in the unexposed, an odds ratio, which is what you get from a case control study, and a hazard ratio, which is what you get from a specific kind of analysis. And this is a pretty great way to quantitatively estimate the risk of an exposed group divided by the estimate of a risk in the unexposed group. And so pull out your phones or your calculators and let's walk through a quick example so you can kind of get a little bit of an instinct for how people do this. So let's say we had a cohort of 200 people, which is a little tiny cohort, but I have to come up with an example to teach. We have 100 of them are exposed and 100 of them are not exposed. And I'm going to tell you this information. 30 of them had disease. 20 of the people who had disease were exposed, and 10 of the people who had disease were not exposed. And then whatever this is, 170 people did not have disease. 80 of them were exposed and 90 of them were not exposed. And so now let's think about what the risk of disease, whatever this disease is, what is the risk of disease among the exposed people? How likely is it? So remember I said risk was a number of people the number of cases over the total number of people who could have gotten disease, right? So what's the risk in the exposed group, right? Well, it's 20% or 0.2. What's the risk in the unexposed group? Oh, shoot, I showed it. All right, so you can see sort of right here that the risk in the exposed group is 0.2 and the risk in the unexposed group is 0.1. You can kind of see these numbers and that makes sense, right? But then what we do, we don't show those in studies. We calculate what's called a risk ratio, and that's the risk in the exposed over the risk in the unexposed. And you can probably look at this and say, well, it's twice as high in the exposed. And that's true, it is, because you put the risk in the exposed divided by the risk in the unexposed, and that gives you 2. So the relative risk is 2. That means it's twice the risk of disease in the exposed group. And so now we get into the language that you might read in papers and you might read in the newspaper. And so the basic interpretation of any of these ratios, a risk ratio, rate ratio, odds ratio, hazard ratio, it actually doesn't really, I mean, there's a lot that does matter within that, but what doesn't matter is how you interpret the basic ratio. If the ratio is 1, that means that there's the same risk in both the exposed and the unexposed group. Right? So if the risk in the exposed group was 20%, but also the risk in the unexposed group was 20%, then the risk ratio would be 1. And so that's what we call null. There's no association. Exposure doesn't cause any more disease. If the risk ratio is higher than 1, like we just got a 2 in my little example, then that means that the exposure, it implies that the exposure group is at greater risk. Right here, if the exposed group was 0.2 and the unexposed group was 0.1, then you might get a relative risk or risk ratio of 2, which means there's more disease in the exposed group, which is maybe a clue that exposure is not so great. You can also get like sort of protective exposure. Think about like in a vitamin or a vaccine or something like that, or mask use during COVID. You might expect that to protect people. Well, the way you would see that is that people who are exposed to the vitamin, exposed to the vaccine, exposed to the mask use would have less risk, 0.1, compared to the unexposed group of 0.2. And here you get a ratio that's lower than 1. So it's, 0.5. Pull out your calculator and check it out. So there's half the risk in the exposed group compared to the unexposed group. And so these ratios are super common in epidemiology and often then dictate how people talk about it in the newspaper. Twice the risk, half the risk, 30% more risk, 20% less risk. These are almost always come from ratios. And you can think about it as if we knew the risk in the exposed and we knew the risk in the unexposed and how we would compare them in this fraction. That was a lot. Most of you still even seem awake. So that's pretty impressive. Willie, I can't tell if you're awake, but I believe that you're awake. You're just off camera.
I have a question, Dr. Costello.
I'm happy to answer
So,
your question.
yeah, what total of research what you did, or what have you been doing it? How much the portion of all this different method of a research what you are doing as an epidemiologist?
Like, which of these methods have I used?
Right.
I've done cohort studies, case control studies, and cross-sectional studies.
Which one is the most kind of proportionally kind of
More
just
common for
Mhm.
me. I've done more cohort studies. I look at big occupational cohort studies of that's my most common. My
Mm.
dissertation when I was in grad school was a case control study. So I really learned epidemiology with the case control study, but I haven't done a case control study since I graduated, and I graduated in 2008. So a while ago now. And then I've been involved in some surveys, some cross-sectional surveys, but my primary main expertise is in retrospective occupational cohort studies.
But wasn't that accurate though? Retro.
Are they accurate?
Yeah.
Is it
Yeah.
Yeah, it's a good study design. Yeah. Yeah, I think it is one of the best study designs. A prospective cohort is a little bit better, but you have to wait so long to get the results that it's a little bit impractical. So a retrospective cohort, the only reason a retrospective cohort might be worse than a prospective cohort is if the researcher did a better job finding out what the exposure was for the cases and the not cases. Like if I knew in my cohort who was already sick and for some reason I was a terrible researcher and I tried really hard to get really accurate exposure assessment for the people I knew was sick and I just didn't really bother for the people who didn't get sick, then I could create a bias. But we don't do that at all. We we have our data sets are really separated from each other. So when I look at exposure, I have no information about what illness people had. I clean up exposure and I make my decisions. And then when I look at who's gotten sick, I make all of those definitions in a separate data set, not with the exposure, and then we combine them and then we look. So, which is because no one wants to do that mistake. So that sort of mistake is really only done if the researcher is being an idiot. Whereas some of the other mistakes that happen in other study designs are not like so obviously researched or they're not as easy to avoid. Like I can just avoid the bias that I don't want to have by being careful. That's easy. That's fine. I'm happy to be careful, no problem. But in the other designs, there are problems that the researcher has no control over, and those can cause really big issues. Or not. I mean, sometimes they're great. When they're great, they're great. They're cheaper. You can get really detailed data. I mean, a really good case control study it can be like absolutely the best way to answer a question.
Great, thank you.
You're welcome. Good luck with all of your research as you get through the summer and you have a question that you think I can answer, you know how to reach me, and I'm happy to answer any questions.
I had it's just a curious question.
Yeah.
Okay. Remember the study, the ecological study you mentioned about Protestants, but someone went
Uh-huh.
back and did like a more individual related. So like what kind of study does that become? Like
Hold on. Am I still sharing my slides or did I stop sharing them? I stopped
yeah,
sharing them.
you stop.
Oh, when someone went in and looked at the individual data, what is it called?
It's what did it evolve into?
It turned into I would say then it turned into it's a good question. Probably a cross-sectional study. They probably looked, but then maybe a cohort study. It depends on when they had the information in time. Like, did they have the rates of the county and then 2 years later the suicide rates? That would be with time in there, so it would be more like a cohort study. That's sort of it's an interesting example because actually the researchers did collect individual information, but they didn't use it in their study because I think they just thought they could make a better point not using it. But they did have it. And so mostly now, if you have individual level data, you would never show it in that group way. I mean, this was 1890s, so like a long time ago. And he really had a very different purpose around like religiosity and social organization, and he was doing something totally different. It just ends up being like a really shining example about how the group level can lead our brains naturally to a different conclusion than if we had the individual
Thank you. I was
level
just
data.
randomly curious.
Yeah. It would be called, I mean, if there was individual level data, then it would be, and there was time, then it would be a cohort. If there was no time and it was all at the same moment, then it would be cross-sectional.
All right, thank you.
You're welcome.
Any, the nurse survey that's still going on from the 1970, still going on?
Not only is it still going on, but that what I described is what they call Nurses Health Study 1. And then in like the 90s, they enrolled a new bunch of nurses who were younger. So Nurses Health Study 1, the nurses were already like 30. And then in 1990, they enrolled, or something like that, they enrolled a bunch of new nurses who were younger, it's like 20, and not all married. And they did a little better job getting like not only white nurses and so they did a better job in 1990, and then they have then now Nurses Health Study 3 really recently, somewhat recently, and Nurses Health Study 3 includes male nurses and just more races, and they've done Nurses Health Study 1 was like older white married women, which is
did they find out any major linked disease only for prevail among the nurse society or community?
The funny thing about that cohort is they're not even terribly interested in the fact that they're nurses for the most part.
Mm.
Like it's more that they are they really have extrapolated to like whatever they find in that group is relates to everybody. It just happens to be that nurses are really great at reporting their information and collecting their own samples, which is true. They are really great at that. There are very few questions that have been, but actually, I would say most of what we know about nutrition and hormone and birth control, we have discovered in the Nurses Health cohort. I had a friend who was a nurse years ago. My first job was at the Veterans Affairs Hospital, and I had a friend who was a nurse there, and she was an older white lady enrolled in Nurses Health 1, and she was part of this little sub-study where every single day for like 3 weeks, she would draw her own blood and spin it down, draw her own urine and spin it down, what she ate, what she wrote. And she was on this like super sub study. And I answered all these questionnaires. And it was something about menopause and hormones and diet or something like that. And but it was like you could not get most people to put in the amount of effort and time. And she had to do all these samples had to be taken before she ate in the morning. And like, I mean, just like super detailed
Detail.
information that takes so much dedication. And so I think like a lot of what they found, they have just, it's like what we know for the general public. There are a few things like one of the first indications that shift work causes breast cancer was also found in the Nurses Health study. But it was actually not a great
Mm.
study, unfortunately. But that doesn't really matter. It was still, these clues, lots of clues have come from the Nurses Health study.
Okay, great, great.
Yeah.
All right, thank you so much.
Yeah, you're welcome.
Very,
Take care,
very
everybody. Nice to see you
interesting.
all. Have a good
Thank you.
summer.